This is episode 60 of the Immunology Podcast, Immunometabolism and Cancer Immunotherapy with Dr. Greg Delgoffe. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Raub. Welcome back to the Immunology Podcast. We have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Greg Delgoffe from the University of Pittsburgh on the podcast to talk about his research on the metabolic contributions to T cell fate and function. We also got our usual roundup of recent highlights in immunology news coming up. But first,
don't forget that IUIS 2023 is coming up. We are looking forward to attending the 18th International Congress of Immunology, which will take place in Cape Town in South Africa between November 27th and December 2nd. Early bird registration rates are available until August 30th, so not that much time. Register at iuis2023.org. I never know. How are you supposed to say it, Jason?
Either one.
Either one? What's the American way?
I think ORG and org are both used, although for .com, it's always .com, not .com. So, I don't know.
Okay, so iuis2023.org.
Speaking of that, we have to plan, like I'm staying an extra day. We got to plan what we're doing if you're staying as well. I booked my tickets, so.
Oh, I have to finish taking care of that. Yeah, we need to go somewhere fun.
Yeah.
If any listeners from Cape Town have any suggestions, just throw them our way.
Yeah, it's an extra day only on my end. So like wine, yes, probably can't make it out to safari, but like I hear Cape Town has great wine country nearby. So
I like that. I mean, I'm not sure. Maybe I got a couple of days extra, but not a lot. I think I'll leave the safari for some other time.
Yeah, I think I go back December 4th. So I have like all of the 3rd to do things.
All right. We need to make it memorable.
Yeah.
So, Jason, happy belated birthday.
Thank you. Yes, it was the big one. I am now 40.
Yeah. How does it feel?
Well, my joints remind me every day I'm middle-aged, but my approach to life is I'm going to slide into my 40s like a bus without brakes and just keep going.
Oh, I mean, the 40s, this is like a roller coaster that only goes up. It gets better. Well, that's what I hear.
Yeah, I don't know.
In reverse.
My body reminds me every day that I don't heal like I used to, which is sad.
Oh, well, I wonder. I can only imagine how it feels, here from my early 30s.
Young in Europe with European healthcare that keeps you nice and well preserved. I know.
I know.
Great.
I know. And all the breaks, another relaxation we get to have, all the vacation days we get. In Europe,
apparently
today's relaxation day.
Yeah, apparently we're airing on relaxation day. I don't know what that means. I don't understand that concept nor that word.
No, me
that
neither.
Where like you do something that's not work as your side gig to help you out. Is that relaxation?
I guess like just adding more tasks to your day because it's just that's what you do. That's what you like to do. I mean, I find that relaxing. I guess. I mean, our listeners could chip in, and if anybody knows what relaxation means, please bring it on. But for now, why don't we relax into some science? What more relaxing thing to do than
That's true. I wish I had a paper on NMR so I could talk about like hydrogen relaxation times and then have a talk about spectra, but we don't, sadly. Although I do have a paper with an X-ray crystal structure. So if we're going to use really bad segues going from NMR structure to crystal structures generally, I guess I have a paper we can talk about. But that has to rank as probably one of the worst segues we've had ever.
Please, just start with the paper.
Put it out of the misery. All right. This first paper here, after the world's worst segue, is microbial host isozyme analyses reveal microbial DPP or DPP. Yep, 2 Ps, 4 as a potential anti-diabetic target. It came out in Science, August 4th. Its first author is Kai Wang. And I think there's multiple first authors here based on the symbols here. So we have Ziwei Zhang, Jing Heng, and Jia Liu are all co-first authors. Last author is Cheng Tao Jiang. This is a bit of immunology. It's more just pure microbiome, but because bugs exist in your gut and they love your immune system, we're covering it. It's also really quite a crazy story that's super cool. All right. So you've probably heard about all these medications people are using, their diabetes medications, but for weight loss lately. So those all act as GLP-1 agonist. They're actually synthetic GLP-1 that you can inject. But there's a whole other set of medications that inhibit something called DPP-4, which is an enzyme that degrades GLP-1s. These are the gliptin drugs. Sitagliptin is one of them used in this paper. But these are all these gliptin drugs. These gliptin drugs are a mainstay of diabetes, but it's been known for a while that some people respond to these drugs and some people don't.
Sorry, Jason. So GLP-1 is a hormone, is a peptide? What exactly?
It's a peptide hormone that regulates insulin sensitivity and glucose tolerance. So more GLP-1 means more glucose tolerance. It also makes you feel fuller, which is why it's used as a weight loss drug now. Wegovy, other drugs, that it's this big thing now is a second benefit. So the synthetic peptide is one set, but before that, there were these old drugs that blocked an enzyme that degraded your endogenous hormone. So that's what this is about. So they kind of don't describe the assay very well and put it in supplemental figures to bury how they do the first part of it, but they do a screen of microbial peptides that are mimetics for human enzymes. So microbial host isozymes. And they do it against standard drug targets or things that we would care about, right? Like, so DPP-4 and a bunch of other things like, enzymes that do A or B matter for human host disease drug. So we're going to do a screen against that. And so they did a plate-based screen, and DPP-4 popped up as one of these big ones that in particular, Bacteroides, which is a clade of bacteria, that big group, but specifically then a specific bacteria in that, which I will get to the name of in a second because it is very hard for me to just straight up pronounce without looking at it. But these Bacteroides species produce a isozyme that works like DPP-4. And then they do a bunch of work, and I'm trying to condense it here. They show that at baseline, the microbial enzyme doesn't have that much effect on the mouse models, which makes sense because it's staying in the gut. But if you give them a high-fat diet, that's known to cause gut leak. And then it gets to the bloodstream, and they see that in mice with a bunch of Bacteroides, and in particular this one species, Bacteroides theta, I'm not going to be able to pronounce this, theta atomicron, so BT as we're going to call it, Bacteroides T, or Bacteroides theta, which produces a lot of this, that it degrades GLP-1 and leads to less glucose tolerance, signs that are worse like diabetes, right? So the high-fat diet leads to the gut leak, lets this enzyme cross and do bad things because it's working like DPP-4 and degrades GLP-1, which is how they found it to begin with. They then do some monoassociation studies. They do germ-free studies. They do antibiotic ablation studies. They do knockouts of the host enzyme and only the bacterial one to, really show that it's the bacterial driving this change. They knock out it in the bacteria, they mutate it, they do all the things that show and really establish through a bunch of very tightly well done work that the bug in a leaky gut, so in a high fat diet, not a regular state, crosses over and will degrade GLP-1 and is responsible for insulin resistance. And compare relative insulin resistance and a baseline host that's the same otherwise, right? The host enzyme not changing. Then they look at people and they get clinical trial data from people who are on this gliptin drug and break it off into high responders and no responders. And what do they find except that the low responders have more of this Bacteroides clade as well as this specific bad bug. And they find more of this enzyme activity in the serum and see that it affects results of the therapy. And then they recapitulate this back in the mouse models. They show that, hey, if I have a mouse with regular amounts of this enzyme and I give them my gliptin, insulin or glucose resistance and sensitivity is improved. But if I throw in a bunch of the microbial one, the drug don't work no more because the drug does not bind to the microbial isozyme. And then they do crystal structures, which is how we got into a really bad segue, and show the structure between the two versions, because they already have the structure of the regular protein in humans. They do one of the drug of the mouse version. They do one with the drug bound to it. And then, because they're not done, they go back and make a molecule that can block the microbial one and not the human isozyme, and then modify it to be even more gut local to drop the bioavailability way down. So it's very little orally bioavailable, but acts only in the intestine, and then show in the mice that it works to treat the problem. Thus creating a potential therapy with very low bioavailability that blockades the microbial enzyme, so it's bound when it translocates and doesn't work. And so then that lets people who would be on the gliptin drug regularly have better success with it. And there you go. Then they drop the mic and sail off into the sunset, because that was a lot of amazing, crazy work.
What a cool story. Man, I mean, everything about it is very interesting. Like starting with the ability for this particular microbial product to modulate our our general metabolism in such a specific and extensive way.
Yeah, the crystal structures between the human and the microbial one are close. They're just not identical, right? But they have the same active site at some of the other moieties that prevent the drug from blocking, but it's actually an isozyme.
Yeah. Man, those microbes, they're screwing with us all the time.
So I'm sure, we'll say it's immunology because a high-fat diet and leaky gut is an inflammatory state. But regardless, this was really cool science, and I'm here for it. And it's affecting your diabetes drugs.
For sure. Well, I mean, it is in the topic of today's conversation about metabolism. So I think perfectly on the right note. I also have, a couple of my papers for today. One of them is, it is immunology, but it's more about the biochemistry behind immunology. So I'm going to start with that one. And this paper was published in Nature, just plain Nature. No, sorry, Science. I'm sorry, it's not Nature, it's Science. Oh, we're gonna get called out. So also a really, really interesting story in Science. And I'm a little bit reluctant to say the title of the paper because it pretty much gives away the whole thing. But human sting is a proton channel. Bum bum bum. First authors, Bingsu Liu and Rebecca Carlson and Ivan Pires and Matteo Gentili. I think they're all first co-authors. And the corresponding authors are Daryl Irvine and Nir Haikohen from the Broad Institute and the Koch Institute in Cambridge, Massachusetts. And as I said, a little bit gave up the main deal, but I think it is a pretty big deal because, STING is a very important enzyme related to our ability to perceive foreign DNA and nucleotides that are not ours. And so STING, which stands for stimulator of interferon genes. So it is a cytoplasmic receptor, which is capable of sensing cyclic dinucleotides, which are derived either from bacteria, as a PAMP, or synthesized by a synthase called C-gas, which makes cyclic GMP-AMP, and this activates STING when it recognizes cytosolic DNA, which can come for example, a virus. So very important sensing molecule. And we know for a fact that activation of STING, which results in a conformational change and a translocation of the protein of the sensor from the cytoplasm to from the endoplasmic reticulum to the Golgi. And then it has multiple effects, particularly activation of the inflammasome, of interferon stimulated genes, and also activation of what is known as non-canonical light chain 3B, which is part of a non-canonical autophagy pathway, which is also important. If you're a cell that is infected by a virus, you want to get rid of yourself. So that's a way of doing it. And of course, as I mentioned, inflammasome activation as well. So we quite understand. So I'm not an expert in STING, but we quite understand like how STING mediates activation of, for example, the interferon regulatory gene activation. It interacts with some certain adapters and certain kinases, just TBK1. We know that, but it's not completely clear how STING activates the inflammasome and how it activates this non-canonical LC3B. So activation, which is through a process called lipidation. And it's not clear how that we know there's certain ATPase related enzymes that are associated to this, but it's not clear how they get the signals to work. And so the authors kind of want to understand how is STING interfere, how is STING starting up this process. So again, not an expert, but basically what is important is that STING in the surface of the Golgi reticulum, it interacts with certain enzymes and generates this lipidation of LC3B, and then this starts this autophagy activation. And one of the things that happens at some point is that there is a transport of proteins. There's an acidification that is important for the lipidation of this protein. And it was never clear how this happens. And so the authors kind of wanted to really look into this closely. And so how do you find out where this protein, how these proteins are being transported across the Golgi membrane? And how do you find if STING is related to it? So what they see is that they act, so there's a bunch of STING activators, and they have models in which they activate STING, and they have these really cool sensors, these proteins that can sense changes in pH in different, and they can direct them to different organelles. So either the different parts of the Golgi or also lysosomes, and using by targeting particular proteins associated with this compartment. And what they see is that in fact both the trans Golgi and the cis medial Golgi do get acidified. So they see a reduction in the pH, which suggests that in fact there is a transfer of protons. And kind of long story short, what they see is that this transport occurs directly through a pore that is contained within STING. And this is not so straightforward. So it's not, as a non-structural biologist, I think, well, how did they not know this? But this is not very self-evident. They started this, they kind of arrived at this conclusion after looking at, understanding that there is a transport of protons, that STING is important for it. And then they have this, I think it's cryo-EM structures or crystal structures, I can't remember, in which they show that the structure has a predicted pore inside, which happens only in the case of an activated STING. And they eventually find a particular compound, a STING agonist, called compound 53, that binds to this particular transmembrane domain that STING has, was kind of predicted to be a pore. And they use this protein to kind of to see if they could kind of use by blocking this pore, whether this changed the function of STING. And so what they say is that when they change, so if they treat, sorry, I'm a little bit confused now because because they say that C53 is a STING agonist, but when they actually treat STING and they activate STING using well-known agonists such as CGAMP or diabz, which are also well-known agonists, then they actually see a reduction of STING, of the pH increase that is observed in the Golgi compartment where STING is activated. So now I just realized that it's a bit confusing now, because basically what they find is that using this, what they conclude is that by using this molecule, also by doing a lot of very smart experiments, which are a little bit long to discuss, they show that indeed it is STING itself that is moving these protons across, and then that this is activating the rest of the pathway that ends up activating the autophagy in STING activated cells. So in the cells that are were activated with STING agonists. So, and they do this also even if they do this in Golgi, so they have this in kind of the endogenous Golgi cells, in full cells, but they also look at it in reconstituting liposomes. So they do like little liposomes in vitro, like outside of the cells, and they put STING on them, and then they show that again, this protein exchange depends on STING not having this pore obstructed by C53. So, and then if you do this, if you prevent this channel activity of STING, then you reduce the lipidation that is the key kind of downstream event that happens and then continues on the activation of autophagy. And this is also important for the activation on inflammasome activation. So in principle, this seems to be a very crucial function of STING, which apparently was not previously determined. So it must have been a really cool thing to discover, because if you're in this field that you didn't know how STING worked in this sense, this must be an exceptionally important piece of information to have. So yeah. So basically, STING has many faces, and as part of the immune system of the cell, recognizing foreign DNA and foreign nucleotides, I think it's very important that we know how it works.
No, I think you're right. I think the fact that it's a hydrogen pore is really interesting, and something that I wonder how many more times this will come up where we really don't understand how these proteins work.
Right?
We keep trying to be good at structural biology, but that seems like a big hole that we didn't know. Pun intended.
Yeah. Boom, boom, boom. So yeah, I mean, it's very complex. It's a lot of structure, a lot of like hardcore biochemistry. It's a cool paper, but I leave it over my head sometimes. But yeah, it's just, I think I'm mostly surprised by that. We know this already? How? But it's true that the STING pathway is not known for so long when you think about it. I don't know, like not even 10 years or 10 years, something like that. It's not
True.
like it's been known for such a long time either.
That's very chill. I also have to thank you, Brenda, because like you probably do in most of your life, you did all the hard work and can let someone in come afterwards with the second part, because you did all the work explaining what STING is. And I also have a paper on STING.
Well, you're welcome.
Thank you.
Happy birthday.
Thank you. So this is the C gas STING drives aging-related inflammation and neurodegeneration. First author is Mohammed Gulen. Last author is Andrea Blaser. This one is in Nature. So this is a fascinating paper. I have to say, when I looked at figure 1, and I'm like, how are they doing this? They have some senescent cell models, like aging cell models, but like figure 1C is in mice, and the timeline of age, it's age months, and it starts at 22 months of age. So they just have to keep old mice around in this lab, and I feel bad for whoever has to do that. That is a rough job. That being said, what they look at is that they have a inhibitor to the STING pathway called H151. And they basically show that with aging, you have increase in inflammatory cytokines in the brain, type 1, TNF, IL-6, IL-1 beta, pick a flavor of ice cream, it's there. And they take young mice, compare it to old mice, and they compare it to old mice with the inhibitor, and they all go down. Like, oh, well, blocking STING seems to help against aging-related inflammation. So they show that, and then they can use inhibitors and look at neurodegenerative staining in the brain and see that, lo and behold, if you get rid of sting, or blockade sting, you have less aging markers in the tissue of the brain as well. And then they really isolate it through some single cell RNA-seq and staining to microglia function. And so they established that it's really these microglia that are changing. They have a change in transcriptional state. They extend it out, and they find that the changes in the microglia then affect other systems. They use an inducible sting model. So they create an inducible tamoxifen-induced sting model, locally, tissue specific to the brain, drive it, turn it on with tamoxifen, cause artificial aging, cause the signaling, see the damage like you see in old mice. So they isolate it that way. They show that the changes in the microglia lead to bystander cell inflammation, neurotoxicity, and impaired memory capacity. But wait, how is STING doing this, right? As you mentioned, STING senses nucleotides, right? So they isolated it to, and they noticed on their staining and electron microscopy, the mitochondria of aged mice in these cells looked damaged, bad. Pick an adjective. Well, damaged mitochondria, at least mitochondrial DNA, and that's what's driving STING. They find higher concentrations of mitochondrial DNA in old mice tissue, which drives STING, which drives inflammation, which leads to neurodegeneration. And I think at the very end, they used a drug that stabilizes the mitochondria, if I remember right here, and reversed it.
What a twist.
It's the mitochondria who did it.
Well, we're going to be talking about mitochondria later today, too.
In the microglia with the DNA.
Betrayed by your own mitochondria. Who would have thought?
Yep.
And what's the compound that they use? Can I have that? Some of that?
Hold on. Let's hear mitochondrial DNA. So it's voltage gated. So they didn't block the damage. They used a VDAC oligomer. So researchers implicated voltage dependent ion channel VDAC13 release the mitochondrial DNA. And so they used VBIT4, an inhibitor of the oligomerization. So the mitochondrial DNA can't get out of the mitochondria when they're funky.
So they kind of, did they use that also to accelerate mitochondrial death to like see?
No, they did it the other way.
Or?
So aged mice leaking mitochondria, they prevented the leak of the mitochondrial DNA out. Separately, they made a mouse that induces STING activation that's inducible to then cause damage. So they showed mitochondria leads to STING, stop the mitochondria from leaking DNA, less STING. They also then turned STING on or off and showed the effects in the brain recapitulated with STING alone. And then it caused bystander damage, all in the microglia, and particularly in the hippocampus.
Okay.
So STING stings you in the brains.
Oh my God. Brilliant.
Look, my jokes are getting older too.
Were you stung by STING?
I am. I definitely, the hippocampus is failing here, folks.
Yeah, that's okay. You're 40 now. I'll be gentle.
I'm senescent.
Oh my God. You're in the prime of your life, Jason. You're in the prime of your life. Okay. So last paper today. No, we already rewrote STING biology. Let me, hit you with another, with the word revolutionary thought. What about antigen agnostic CD8 cytotoxicity? How do you feel about that?
I'm mild to moderately intrigued, but could be moderately to severely intrigued with further information.
So, next paper, CD8 T cells maintain killing of MHC1 negative tumor cell through the NKG2DL axis. Yeah, this doesn't sound so exciting, but it's actually quite exciting. I like this paper. First authors Emily Lerner and Carolina Voronica from the lab of Peter Fecci at Duke University. And again, I think it's a really cool story that starts with a kind of very simple experiment. So I think those are the best. CD8 cells have TCRs, conventional wisdom say they pick up their antigen on, for example, we're working here with a cancer setting, tumor cells, CD8 cells, find their target through their TCR and attack cells expressing the target antigen on the right MHC. But we also know that amongst the many things that tumor cells do to try to fend off or the immune system is getting rid of their MHC1 expression, often through mutation and loss of expression of beta 2M, which is this microglobulin that is important for the assembly of the MHC class 1 molecules. And that conventional wisdom would say, well, then these cells become kind of invisible to the adaptive immune system in the form of CD8 cells, because if they cannot see them through the TCR, how on earth are they going to find these tumor cells? And basically, this work does is show that there is an alternative way, which is a mechanism that we already knew could happen, but we saw it always associated to NK, to natural killer cells, which is the recognition of tumor, so MHC or beta 2M knockout cells, so non-expressing MHC class 1, through ligands of these receptor called NKG2D. So we know that NK cells use this pathway. No controversies here, but as I'm going to describe briefly today, the authors of this paper show that it's not only NK cells that can benefit from this non-traditional recognition. So they work with a model of tumors that are this CT2A glioma model, and what they do is they started studying the effect of checkpoint inhibition, anti-PD-1 and 4-1BB stimulation as a type of immunotherapy. And what I see is that even even if you have tumors that are beta 2M knockout, you still have a robust recognition and rejection of the tumor upon checkpoint inhibition. And so that was they thought, I guess the idea when they started this experiment, they thought they would have less. And what they see is that, importantly, so there's a lot of experiments. They are very thorough, but I'm going to kind of highlight the high level things here. They show that this recognition depends not on NK cells, which would be probably everybody's first thought, but depends on CD8 cells. And when they dig a little bit deeper, they see that actually the CD8 cells don't even need to be tumor specific to mediate this response to checkpoint inhibition. It's just enough so that you have CD8 cells that are activated by someone in the tumor. And they show that often that someone will be macrophages that are cross-presenting. So you need to have the antigen somewhere. So those cells if you have, for example, these CD2A specific cell, so this tumor cells have a TRP2, which is there's a specific TCR that recognizes. So you still need to have T cells that there's antigen that they can be activated against, but then these T cells don't necessarily have to go attack the cell expressing the antigen. Of course, I guess my first thought was like, well, that's problematic. How do we keep T cells from just attacking everything? And apparently they don't do this. The effect is quite localized. The T cells do need to be activated like right there by, for example, a macrophage. And they eventually, what they show is that this recognition is mediated by ligands of this, traditionally molecule was traditionally associated to NK function, which is very important for NK function, NKG2D. And they see that actually these tumors, because they overexpress so much of these ligands for this, such as RAE1, H60, MALT1. These are all ligands for this NKG2D. this is sufficient for activated cells, activated CD8 cells, which are expressing NKG2D, which is known as an activation marker. It's known as, was usually associated as a co-stimulator in CD8 cells. This is sufficient to redirect the cytotoxic activity of the CD8s onto the tumor in an TCR agnostic manner. And that, even though this is kind of unspecific, most healthy tissue gets spared because they don't overexpress ligands for this particular receptor. So they do a lot of they really work through in mouse models, and they also gonna go for the human setting in which they also look into human cells, and they see that it can also be applied to assisting in vitro of human cells. And they also look into data associated to human cancers, and they also see that there was always a little bit of controversy whether beta-2M is a bad thing, is a good thing. It was not clear from the human data, and this might be a reason why it's so murky to see the effect of MHC1 knockout or non-expression, because they might be the CD8 cells might be circumventing the whole thing. So I thought it was really cool. So again, I think it was a little bit mind blown. And this was published in Nature Cancer, by the way. So really cool work, and also very elegant. I love this. I love this artist works in which you have very specific, like the experiments are all very nice, and the story is really well told. So kudos to the authors as always.
Yeah, I wonder what this means also for autoimmunity, right? If you just have some CD8 cells that are hanging out a little activated.
Yeah. Well, I mean, what they say, so they do think that they do an experiment with I think it was just regular what's the word, fibroblasts, and they don't get attacked because they don't express enough of the ligands. But yeah, I don't know, I think it does give you another thing to think about when you're thinking about immunity. Yeah. Or at least bystander killing, bystander effects on bystander cells.
Yeah, makes sense. Interesting. All right. Well, we're going to actually talk more about T cells here soon with Dr. Greg Delgoffe at the University of Pittsburgh. But before we do that, you can decorate your lab with a Nature's Protocols wall chart. It outlines the production of therapeutic CAR T cells from apheresis collection and T cell enrichment to gene modification, expansion, delivery. Request a free copy of the wall chart at STEMCELL Technologies T Cell Therapy Resource Center by visiting stemcell.com/T-cell-therapy.
Well, hi everyone. To today's interview, we are joined by Dr. Greg Delgoffe. He's associate professor at the University of Pittsburgh and director of the Tumor Microenvironment Center at the UPMC Hillman Cancer Center in Pittsburgh. And he's going to be talking to us today, I assume, about his research on immunometabolism and how cancer and the immune system interact with each other through metabolic pathways, and how that informs or helps us understand why cancer happens and what to do about it. Dr. Delgoffe, thank you so much for joining. I'm a big fan, so very excited to talk to you today.
Yeah, thanks for having me on. It's a pleasure to be invited, and a pleasure to share the work that we're doing and just kind of to talk science.
So we tossed the coin with Jason here, and I got first dibs on today's conversation. I guess I'd like to start with maybe a little introduction. Immunometabolism, or understanding how the metabolic properties of, in this case, cancer, or maybe other many diseases, influences how the immune system responds to this particular situation. I think is fascinating, and it's a field that has really exploded. I would say in the last 10 years, it's been really a huge increase in our understanding. So maybe would you like to talk to us or give us a little introduction on why do we care about metabolism, and what do we, when we say metabolism, because I think it's a very wide, a very encompassing word. What do we think about metabolism, and why do we care about it in immune cells and maybe T cells in particular?
Yeah, great. I'm happy to do that, and it's something I love talking about because in my my past, I've always been interested in the regulation of immunity. That's always been my number one kind of interest is how is the immune system regulated, especially T cells. And when I was being trained, it was always about signal 1 and 2, right? You need antigen, you need co-stimulation. Later on, we added cytokines as signal 3, and but the point is that especially a T cell, they have to operate in all these different environments, right? That in that nursery of the lymph nodes, but most importantly, they need to be able to infiltrate all the tissues to do their job. And while they get different cytokines and different other cell surface cues, they're also in different metabolic environments. And so it makes sense that they would have evolved to sense those metabolites. And that's really how I got started in this field, as I studied the role of a nutrient sensor, mTOR, in how T cells make decisions. And that's what I cut my teeth on in graduate school, was really understanding that before we could really interrogate metabolism at any kind of cellular level. We were just really saying, if you change the nutrient sensing capacity of a T cell, you change how it functions independently of all the other things, of all the other signals. And I think that's something that we're really starting to appreciate now, is that metabolism is this kind of primordial means by which cells are regulated. And that makes a big difference. And so we have to be thinking, I think you brought up a really interesting point at the onset, which is metabolism is everything. It's like life, right? So it's kind of hard to really think about, what do we mean when we say immunometabolism? And I think it's multifactorial, right? Are there nutrients in the environment locally that are changed? Is it how the cell intrinsically changes its ability to sense those nutrients? Or is it how the metabolites inside the cell, how their catabolism or just even kind of moving around or exchange from one type of carbon to another, does that alter the way that the cell performs a particular function? And we think this is super, super relevant in immunity.
So we know about the Warburg effect with cancer, right? But I think we're also appreciating it's more simplistic than what's really going on. You talk about how cancer can alter the milieu of the nutrients that are available for the tumor cells. Now, when I think, or for the immune cells, right, to fight off the cancer. Now, maybe my impression's wrong. When I hear Warburg effect, it's like, oh, glucose is being consumed by the cancer. Well, great. That means everything else is available for the immune cells. Is it also that the immune cells want the same glucose, or is it actually that much more complicated than that? And then maybe they want something else and they're actually changing the profile more than just hungry hippo for glucose. We all learn.
Yeah, no. So this is a great question. It's something that definitely needs to be. It's dogma that needs to be fixed in our field, right? So at the beginning, it was tumors consume glucose, they make lactate. That's it. That's the issue, right? And so what's interesting is, like, we know Warburg metabolism occurs. The important thing to remember about Warburg metabolism in any cell type is that it doesn't mean it's not a switch. Right? It's not that the cell uses its mitochondrion and then it goes Warburg and then it doesn't use its mitochondria and it ferments glucose. I think that's one of the misconceptions. It's that it also does the glucose to lactate thing, but it's still using its mitochondria. So it's not a switch from one thing to another. It's rather the glucose fermentation turns on, but the mitochondria are still doing all their stuff. So when a tumor is fermenting glucose to lactate, it's still also consuming all that stuff in the mitochondrion. Which you bring up another point about the hungry hippos, right? I think it's always, it's very comforting to think about it like a land grab, right? The T cells, they're also doing Warburg as well. We've known about that since the early 70s, actually. That if you before we even knew about T cells, really, to speak of, we knew that if you added PHA to PBMCs, they started to do Warburg metabolism as well. But intriguingly, at first it was like, oh yeah, glucose is in competition. The T cells come in, they want glucose. The tumors are consuming glucose, and that's it. There's not enough left for the T cells. What we're learning is that that's a very simplistic view of what's going on. And that's if you actually measure the amount of glucose that's inside of a tumor, so this is called interstitial fluid. This is the juice of the tumor. And you can get the juice of the tumor quite easily by just squeezing it, spinning it out. And it's the straw colored fluid that kind of comes out. And it's kind of the milieu, right? And we've done a lot recently with interstitial fluid and how that works. But the short answer is the glucose is not that low. It's lower than serum, but it's not like there's none there. But what really is the big issue, and I can't stress this enough. I've even thought about the tumor like a waste, like a desert, right? There's nothing there, right? So the T cells come in, and it's like Lawrence of Arabia. There's just soldiers marching in the desert by themselves, and they don't have anything to eat, and that's it. But I think our views on this have really changed, which is that it's not a desert. It's not that there's nothing there. It's that the tumor is not just consuming stuff, it's also producing stuff. All of the catabolites of all of those essential nutrients, glucose into lactate, tryptophan into kynurenine, or any other, frankly, any other tryptophan catabolites. The oxygen being converted into reactive oxygen species, all of these kind of byproducts, that's the real nasty stuff. It's those things build up, right? And that those things can be toxic or at least immunomodulatory. And so it's not like a desert, it's kind of like a wasteland that our soldiers are fighting in, so to speak. And I think that's a really, really critical kind of not new, but newish kind of look at this cancer immune metabolic interaction in that environment.
Yeah, so there's not only like a lack of certain nutrients, there's a lot of metabolites that are interfering with their function. And this brings you also to a lot of some of your also early papers, and some of the work in your lab has looked into trying to understand the difference between how different subsets of T cells respond different to this wasteland. And I think in particular, there's this idea of the dichotomy between effector T cells and regulatory T cells that seem to have some kind of differential metabolic program, which might explain why they find themselves in such large numbers in tumor microenvironment often, and how do they survive. And I think as a Treg enthusiast myself, I believe that they are, because they're already born different, they're already going to develop different from the very beginning, well, from the thymus. But I think that they already have this program going on that clearly is adapted to different realities. So what do we understand now about how different T cell subsets seem to be adapted to different conditions, and what are we doing with that information?
Yeah, great question. And basically the foundation of everything I do in my laboratory, right, is I kind of think about how these different subsets think about how do they respond differently to different metabolic environments, right? And so if we kind of break it all down, right, and maybe beyond even effector versus regulatory, but let's say like long-lived T cells, like memory or naive T cells. Let's think about those like really, really hot active cells like effector T cells. Then let's think about the regulatory cells, right? The ones that are acting kind of always in opposition to the effectors. And then you've got the exhausted cells, right? Which we can talk about a little bit about as well, which are kind of the antithesis of the memory cell. Like they are short-lived and poorly functional, right? And yes, we think about how these cells change their kind of intrinsic biology to survive. But the especially the effector regulatory dichotomy, I think is really interesting because your Treg cells, and I love these cells. I think that they are very fascinating because of how stable, how epigenetically stable they are, how metabolically distinct they are, and how potent really they can be at suppressing immune responses. But the one thing that is really interesting about these cells is that they have to be working when all the other cells are not, right? When you're at a resting state, if you will, all those cells are being held in check by Treg cells. Like a third, beautiful story from a few years ago, not from our lab, but beautiful paper. When you deplete Treg cells, about a third of your CD4 T cells immediately spontaneously activate, right? So this is really interesting. These cells are always really working. And so it makes sense that their metabolic proclivities may be a little bit different. And so I really took this, came in from the cancer angle, because the one thing I could never write, just kind of break in my mind, was that if this environment is so bad, right? How come when I look at a Treg inside of a tumor, it's like 90% of these cells are in S phase, right? They are cranking, right? They're turning over, they are going wild. So clearly they're able to have a profile that allows them to thrive, and that this place is not bad for all T cells, just for some types of T cells. And that's what led us to really undertake trying to figure out what their metabolic profile is, especially in cancer, not necessarily because, not a priori, we weren't really thinking we're going to make cancer therapeutics, but really more that cancer is a great model to study Tregs because they're so active, they're so powerful, and they're so important in that state. And so we really started to go down and understand their metabolism. And basically what we found is they thrive on the catabolites of tumors. Those pieces, the products of tumor cell metabolism were things that Treg cells had evolved to be able to eat on their own, and actually preferred. When we deny them this, they slow down and they kind of become dysfunctional the same way that all the other T cells do, and they stop suppressing, and we can really leverage this within that environment. I know that was verbose, but that's sometimes what you get from me.
So why do you think they evolved that way? Specifically, like they obviously didn't evolve so that we could have more cancer. That seems a little silly. So there has to be some unhappy accident here leading to Tregs preferring these metabolites, but that was why it did it to begin with. Do we know that? Are we still figuring that out? Like, what's the reason they would want that?
Yeah, we're still figuring. Metabolites? We're still figuring it out. But there's been a number of we posit a few things, and so I'll wax poetic for a second. But we've also been kind of backed up by a number of other studies. But I couldn't agree more. We did not evolve Tregs to give us to give us cancer, and this axis did not exist for this. But so one of the major metabolites we found in the paper in this first story was lactate. That Tregs can really utilize lactates. They thrive in lactic acid rich conditions, and so we really, this is where we really got started. But then we started thinking, well, where else is lactate rich? Right, in the body. And so there's adipose tissue. We know that Treg cells are very enriched in adipose tissue, in damaged muscle. That's where you get a lot of lactates. These are all places that you don't want a lot of immune activity. You don't want scarification of your muscle. You don't want any damage. You don't want any inflammation in your adipose tissue. In almost all immunologically, classically immunologically privileged sites, are high areas of lactic acid concentration, including in your brain. Your brain is a huge is very lactate rich. But the other one that we're very much interested in now is in the gut. Lactate is produced by the commensal microbiome. It is produced in the hypoxic niches of the colon. And so my, gun to my head, I'm guessing it probably evolved first to promote tolerance to commensal bugs, and that metabolic interaction was most likely exploited by tumors to maintain tolerance.
That's a very intriguing theory or proposal. I like it. I know Jason does. See, we're back to microbiome, Jason.
It's inevitable.
You can't get away from it now,
Yeah. I hadn't thought about that, but it's a very, very interesting. And I guess also it would make sense that given that often Tregs will be needed in sites of inflammation, if you have active inflammation, I assume you will probably also find lactate in this circumstances. Might not have been the evolutionary pressure, but that's probably also a welcome side effect of having these cells surviving in this environment.
Yeah, absolutely. We've actually really explored this connection, right? Because one of a very good potential source of lactate is the effector T cell that is being suppressed by the Treg. So we've actually really explored now in follow-up studies that are not published yet, really understanding if all there's no tumor, there's no tissue, it's just a conventional T cell and a Treg sitting next to one another. But the Treg can't sense the lactate from the conventional T cell, what happens? And we're learning that there's a component of the Treg cell program of its signature that is derived even from the lactate coming from its target, the suppressed T cell. And so it's a lot of stuff we're exploring.
So I want to flip this a little. Therapy. I'm a practical person. How can you use this therapeutically to say, let me caveman this. Too much lactate bad because it likes the Tregs and then tumors don't die. I'll pick something. Or too much glucose, or whatever. Is there actual ways that we can alter our metabolic milieu to have a shift on this? Is this something, for instance, that diet can affect, or do you put a lactase as an enzyme in people's serum so it gobbles it all up? Like, I don't know. Like, those are two different ends of the stream here.
No, this is a great thought, and this is something that we talk about, we think about all the time. So, first, the cancer field, right? You rewind, 25, 30 years. This was all the rage. We're going to cure cancer by targeting its metabolism. But and there was actually several trials actually of 2-deoxyglucose. But it was all zero sum. Nothing ended well. And because it worked really well in a xenograft system, right? Tumor by itself on an immunodeficient animal, treat it. Yeah, tumor got smaller. You put it into a person, dose limiting toxicities. And of course, you're inhibiting the immune system and also all glycosylation, right? So very, very tough to target. But that being said, the field of metabolic diseases, metabolic syndrome, type 2 diabetes, obesity has produced a cornucopia of metabolic modulators that many have been thought about for cancer therapeutics, many have not, many have been overlooked because they're not marketable. But so when I started my group, we didn't have a lot of money, so we picked a bunch of cheap drugs, lots of things that we could use. One of them was metformin. Metformin has this long history of anti-cancer, anti-aging kind of stuff. But it turns out that a lot of the effects of metformin on cancer actually depend on the immune system. And so we really started exploring this and understanding that this metabolic modulator, it's a very strong modulator. It's not a very strong drug, but people take grams of it. It's pretty safe, but it could actually change the tumor microenvironment in a good way, and mostly by reducing hypoxia and changing the metabolism of the tumor cells, which itself wasn't enough. We proved that with 2DG. You can't just turn down the metabolism of tumor cells, but it's synergized really well with immunotherapy. When you give that immune boost like anti-PD-1 or anti-CTLA-4, then you would see something. And so this actually led us to a couple of clinical trials that we're running here in Pittsburgh, and there's many other trials running across the world combining these two drugs together. I think that's a pretty sledgehammery approach. And there are effects of the metformin on the immune system that have been well documented. So I don't think but it is telling us something, and our academic interest in looking at the samples from this trial has really produced a lot of new hypotheses, and the clinical results are nothing to sniff at either. But I don't think it ends there with just drugs. I don't think it's like a magic where there's going to be some sort of magic drug that targets some sort of perfect pathway that hits the tumor and not the immune system. This is really where cellular therapies come in my opinion. Is it is part and parcel of immunotherapy, of immuno-oncology, to grow T cells ex vivo, engineer them to see cancer cells, and put them back in. Right. And in the CAR T cell space, in expanded TIL, in making TCR transgenic cells for therapy. So while we're putting in something for to make them cancer specific, why not put something in as well that also improves their metabolism? And in that way, if we can put in a genetic program or an epigenetic program into those cells that allows them to maintain or enforce a better, more tumor, anti-tumor promoting metabolic profile, well, then that would be specific to those T cells, right? And then it wouldn't affect the global environment, and we wouldn't have to worry about off-target effects. So we did this, pretty early on. We used a couple of genes that were involved in mitochondrial health and quality, the generation of new mitochondria, a molecule called PGC1 alpha, which we first looked at in mouse TCR transgenic T cells. And we've now translated that into human CAR T cells. But this has now been really part and parcel of a lot of things that we're doing in the laboratory. Is what are the metabolic programs that we can enforce in a cellular therapy that at least endow those cells with a distinct and improved metabolic profile for anti-cancer immunity.
This brings me to maybe another, you mentioned it before, but the other side of the coin, when you're talking about the traditional view of cell metabolism, you think of glycolysis on the one hand and mitochondrial function on the other hand. And I think our understanding of the role of mitochondria has evolved a lot. They're not just, finishing up the job. They are actually making all this biosynthetic intermediaries. They're, being their hub for a lot of these metabolic processes. And so when it comes to do the function of or like the right mitochondria, or how does the form, the shape of a mitochondria affect this that you described, the ability of a T cell to survive? What do we know about that?
Yeah, that's great. A great question. And honestly, we are still learning a lot. And it's mainly because the tools that we use that have been traditionally used have been, applied in cell lines and cancer cells and things where, when you're really saying, oh, well, what about exhausted T cells? What about memory T cells? Well, can you do that analysis with the 500 that you get? It's tough. But that being said, we have learned quite a bit. A lot of really great work from not just from our group, but from Erica Pierce, from Sukek, really lovely sets of work understanding some of this. But the mitochondria are not just burning things for energy, they're producing things as well. And a lot of that chemistry is involved in what their intrinsic proteome is like. Remember, they have their own genome, so they're producing their own sets of intermediates that are going to do chemistry, and then they import a lot, right, from the nucleus. So there's that kind of, what are those genetic programs, transcriptomic programs that make a mitochondrion work? But there's a lot of morphology that's involved in the cellular cues that bring this in. So the kind of lone wolf mitochondria, those are the ones, the small kind of punctate, we call them fizzed mitochondria. They do fission, and so they divide. But the ones that provide, that are left, they have these kind of loose ultrastructure. And that means that they don't have a lot of space to do chemistry, right? In mitochondria, the reason why they're all folded up is because it's all that surface area is what they need to do all of this membrane-bound chemistry, right? And so a more complex mitochondrion, right? And it's usually one that is fused with others, and they form these kind of spaghetti-like noodly structures. Those have this high degree of surface area. And so what that means is that they become engines that are a little bit more fuel efficient, right? It's not so much that they're better at burning carbon, but they can do oxphos, they can perform the TCA cycle, and they become efficient. And so when they're efficient, that also means that they can free up more of their time, more of themselves to be part of that biosynthetic step of all of this. And what we're learning now in the lab is just how productive mitochondria are. They produce so much that the cell can use. And if you isolate mitochondria from different subsets of T cells, if you get them from the long-lived cells like memory cells, these mitochondria are far more in that very complex networked stage and produce a lot of biosynthetic intermediates. Whereas if you get the ones that are more exhausted, they're all fizzed, they're small, they're malformed, and they're just producing a lot of ROS, and all of the flavors in between. And so mitochondria are incredibly important for changing the rest of the cell's biology. And we think that is in the level of making lipids, so that you can form membranes, making certain kinds of amino acids that you have to synthesize inside the cell, and probably not most importantly, but definitely one thing that we're really interested in now is in altering the heritable epigenome. Is that all of those changes to the epigenome are metabolic in nature and are produced primarily within the mitochondria.
So, mitochondria drugs?
Yeah, I mean, I think that there's a lot of different ways that we can play around with modulating mitochondria. There are, I mean, and in fact, there's actually a lot of interest in mitochondrial transfusions, right? Actually manufacturing in cell lines or in patient-derived samples, healthy mitochondria, right? And then transplanting them into things like therapeutic T cells, and rendering them more long-lived, essentially and you can spin mitochondria into cells. You can coat them in hydrophobic, stuff to get them in. You can tag them with cell-penetrating peptides. So there's and that's actually a big interest in the aging field, right? And the anti-aging kind of thing is like, can we essentially transfuse people with mitochondria to improve their aspects of aging, which indeed is also linked to defects in mitochondrial function over time. Beautiful data actually from a couple of years ago as a science paper. They deleted a critical mitochondrial transcription factor only in T cells. Okay, and that alone produced an aging phenotype. Only if your T cells became mitochondrially deficient. So there's a lot of interest in understanding this. But in terms of thinking about things in cancer, it's the real question about mitochondrial drugs is, how do you hit only the mitochondria of certain cells that you want to target? And I think that's something that we need to really keep thinking about.
Fascinating. I did not know you could transplant mitochondria, and I'm not surprised the aging people are working on it. Sounds that you can spin mitochondria in cells, or very intriguing. I guess you could do that if you're expanding some T cells ex vivo. You could just give them a mitochondrial spin. But I guess you can also use some of there's a bunch of known promoters of mitochondrial biogenesis, I guess. But
yep.
We could continue talking about this metabolism until we age, but I think
we become exhausted.
We're in science. We're all exhausted. We're all terminally exhausted, I'm pretty sure. But not
terminally, just chronically. we keep at it. We just keep at it. But so we like to ask our guests a different question, like something outside maybe of their scientific endeavors to get them to know a little bit better. And I think I'm going to ask you a question that is pretty much a classic in the podcast, which is, if you were not a scientist, what would you be?
Yeah, I think about this a lot, right? Because actually Peter Medawar, who I love, he's a Nobel laureate, but most importantly, card-carrying immunologist as well. But probably wrote the most advice to people about how to be a scientist. But he was very obsessed with the idea that scientist was a phenotype, not a career, right? And that most scientists could very well do other things. I really thought about this question a little bit. I've thought about it my entire life, but honestly, if I had to do something else, I would probably work in a kitchen. I'd probably be like in the culinary space. If you've watched The Bear, it's striking just how much a kitchen operates like a lab. How everybody has their jobs. You're trying to pull everything together at the last minute. You've got your PI types, you've got your postdoc types, you've got your technician types, you've got your students. But it's something that I do for fun, especially now because I'm on this side of the desk now. I'm not doing any science anymore at the bench. So going home and cooking and trying to get a meal together all at the same time and getting everything just perfect, my mise en place all set to go. This is something that is very passionate to me. I'm not awful at it. So I like to think that in another life, I got a food truck somewhere, or maybe I'm running a kitchen or something like that. But it's something that I like to do, but it's also something that kind of tickles my, need to manipulate and come up with something that is ultimately palatable. And so I think that's probably what I ended up doing.
So what's your favorite Food Network show?
Oh man, good question. I'm between two, I'll have to say. Chopped, I think, was one of the best shows on television. It is
Jason approves.
It's been on forever now, so I don't watch as much as I used to, but I just seeing how people work with that.
But you need to give us a description of the show.
Oh yeah, sure. So so Chopped. So what it is they have 4 chefs, up and coming type chefs, and they have a basket, and they open up the basket, and it's got 4 random ingredients in it. And some of them are wacky, like gummy candy and a pig's head and like some vegetable you've never heard of, and
then a brioche bun, right?
Like exactly. And then they have got like 30 minutes to make a meal, right? With everything else at the pantry in their disposal that has to feature the ingredients. And then they get judged by 3 well-known celebrity chefs. And then they decide how it looks. And there's just something about the like impromptu nature of it's extremely entertaining television. And so then they compete for stuff, And
it's what happens in culinary school is one of your final exams. And like there's 3 rounds, it's elimination. So there's 4 chefs at the beginning, final rounds 2, appetizer, entree, dessert, pass through. So you only get last 2 people for dessert. But I also enjoy it because for many years at holiday time or Christmas time at my mother-in-law's house, they would like have some stuff in their fridge and then forget to plan. And then they'd go out and wrap gifts for charity. And they'd leave me at the house and say, Jason, we have a chicken breast, some frozen vegetables, a brioche bun, and whatever spices are in our cabinet. Make Christmas dinner.
I love it.
Which I started appreciating more and more after watching like seasons, 1 through 15 of Chopped.
Yeah.
Oh my God.
Yeah. So absolutely love the show. And then there's this lighthearted talk show on Saturday mornings, The Kitchen, where they just make food and talk, and it's great. So I love Food Network. It's where most of our cable, it's the only reason why we have cable is to watch Food Network. So
I have to say, working in the kitchen or being a chef or cooking is a very popular choice amongst our guests who get this question. There's clearly a pattern. I think this is cheating by now. I think it's just it became the obvious answer because there's as you mentioned, there's so much in common between the bench and the kitchen counter, I guess, that I think we're not going to allow our guests to say that. You guys
that's not fair. Now to come up with another answer on the spot, you're saying, or do I have to?
No, that's fine. This is a new policy we're going to start.
Right,
if you were not a scientist or a chef, what would you be? No, but
good. I like that.
But it's been great talking to you. I just love this subject. And also was great to hear it from you. Your enthusiasm is contagious, and it was. I wish you all the best with continuing this research. Jason, any final comments?
No, just a pleasure, and I'm now reinvigorated and no longer exhausted.
Same for me as well. Thank you so much, guys. I really appreciate the invitation, and this was a lot of fun.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview Roundup Papers. You can also reach out to us on Twitter at Immunopodcast or via email at info@immunologypodcast.com if you have feedback or you would like to suggest guests. See you next time.